My name is Don Sakaguchi, and I'm a professor in the Department of Genetics, Development, and Cell Biology here at Iowa State University. And my lab is involved in developing experimental strategies for rescue and repair of the damaged nervous system. We're developing a biodegradable conduit, a multifunctional conduit that can be used to stimulate and help facilitate peripheral nerve regeneration. We first create a film, a thin film that is micro-patterned with grooves. And we then take this device that has been seeded with these different cell types and we implant them in a peripheral nerve model of traumatic nerve injury in a rat. It's important that the device that we're developing is biodegradable, that it supports cell growth, and that the animal will tolerate its presence within its body during the implantation procedures. We want to culture our cells in a relatively native type of environment. So we compare different types of extracellular matrix molecules. So for example, fibronectin, collagen, laminin, or different matrices that are composed of different constituents. And so that allows us to compare these different types of substrates and the growth properties of the cells. Before having the HCS system in my lab, the experiments were extremely time consuming. We would set up a culture, and we would set it on the microscope, identify a region of interest, and hope that that region became interesting at the end of 2 to 3 days of time-lapse imaging. But often, as you might expect, the site that we had selected turned out to be not a particularly interesting site, and so those experiments would be a bust. The HCS system that we have is an ImageXpress Micro. With the HCS system in a routine experiment, we might use a 96 well plate and perhaps culture in about 60 wells and image 2 sites per well. And at the end of 4 days of culturing and doing time-lapse imaging, we'll generate about 275,000 images. Having a high-content screening system, it just really allowed us to screen large populations of cells, multiple populations of cells under multiple parameters to identify the populations that we're most interested in pursuing. It was a bit overwhelming in the beginning because of the amount of data that we would generate. But it's really refreshing to know that we have all of this information available for us for our future analysis of the different parameters that we're interested in terms of cell behaviors and cell function. So my lab is the only lab here at Iowa State that has an HCS system. And because of that, it's creating new opportunities with colleagues of mine for collaborative projects. Over the next several years, we plan on continuing studies focused on developing strategies for repair of the damaged nervous system. We'll continue genetic modifications of these different populations of cells as delivery vehicles for these therapeutic factors that we're most interested in.